Hey there, humans! In today's class, we're going to look at restriction enzymes. You can also call them restriction endonuclease. Now, these enzymes that we're going to talk about, they have one very specific function, and their function is to cut DNA. When they cut DNA, they look for very specific base pair sequences. So we'll take a look at that in a couple of minutes, but the reason why we use restriction enzymes is to create recombinant DNA. This is where you take two samples of DNA. They don't need to be from the same species, but you want to combine them together to make something new. Recombinants also work in a very similar manner. Okay, naturally, when we're recombining genes here and there, those genes are basically DNA sequences that get recombined in different locations within the same individual. Now, the other thing that we can also use to make is DNA fingerprinting. DNA fingerprinting requires DNA fragments. So in order to get those fragments, we basically use restriction enzymes. There are two types of restriction enzymes that I'm going to introduce to you today. The first restriction enzyme is called HindE3. Okay, it kind of looks like Hind3, but it's actually pronounced HindE3. The other one is called EcoR1. So these two restriction enzymes, they do the same job, but they cut at different places along the DNA chain. So HindE3 looks for a very specific pattern. So here you have a DNA sequence. We're focusing on this middle section, so this portion of the gene. And once they find this specific sequence, it will make a zigzag kind of cut. Once it makes the cut, it will create two sets of sticky ends. These ends here basically can reattach themselves to other DNA that have sticky ends, or if you just separate them and don't attach them with anything else, they just become DNA fragments. So over on the right side, we have EcoR1. So again, they're enzymes just like HindE3, but they will cut DNA at different places. In this diagram, you have two different DNA molecules. Okay, so let's say that this one here is from one organism and this is from a different organism, okay, from a different species. We apply the same restriction enzyme, and the restriction enzyme will cut at a very specific area of DNA, again, creating sticky ends. But these sticky ends do not, like the sticky ends on the right side is from a different organism from the sticky ends from the left side. But because they're cut from the same restriction enzyme, they can be recombined together. So this is a really easy way for us to insert genes into the genome of a different organism. Okay, and basically we kind of use this in genetic engineering or genetically modifying an organism. Okay, so restriction enzymes do have some very important uses. Today we're going to focus on how it's going to be applied to DNA fingerprinting. So we want the restriction enzymes to make DNA fragments. So you're going to need this handout here. If you don't have this handout, I've included a PDF copy so that you can download. Once you have this handout out in front of you, you can follow along what I do here. Okay, so we're going to take a look at EcoR1 first. EcoR1 will cut DNA at this specific base sequence. Now, it's easier to just read the top portion. The bottom portion simply just matches what bases I have at the top via Chargaff's rule. So when I find a sequence like this, GAATTC, in a long series of DNA, the cut is first made between the G and the A. Okay, so the bonds here are cut by the restriction enzyme, and then the restriction enzyme goes and it cuts all the other hydrogen bonds that exist between A and T, T and A, T and A, and then finally it ends up cutting the bonds between this adenine and guanine on the second strand. So it makes a zigzag cut, but it will only cut along this specific base sequence. So if we look at the example below, we have a long chain of bases, okay, DNA bases. If we scan through the entire length, okay, and again, I'm only focusing on the top, okay, we're going to look for GAA-TTC. So GAA-TTC, once we find it, GAA-TTC, so it's right here. There's one sequence here. We're going to make a cut between the G and the A. It's going to end between the A and the G, and it makes a zigzag cut like that. Okay, so what I've done now is on the left side I have one fragment, on the right side I have a much larger fragment. So I'll continue along the chain and look for more GAA-TTC, and I find another one here. So cut between the G and the A, cut between the A and the G, and then cut in between. Okay, I continue scanning, and I do not see any more GAA-TTC. So that means I have 3 fragments. This one here on the left, the one in the middle, and the larger one at the end. Okay. How long is each fragment? In order to figure that out, we have to count the number of bases that are at the beginning to the end. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So the first fragment is 14 bases long. The second fragment, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. Okay, so it's 19 bases. And then the last one, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26. Okay, so 26 bases long. Okay, so EcoR1 creates a very specific number of fragments. The fragment sizes are specific to this particular DNA molecule. If we use a different restriction enzyme on exactly the same DNA, we would have different fragment numbers and more likely different length of fragments. So let's try that in the second example here. So I'm going to scroll down to Hein 3. I'm just going to change the color of my pen here. So Hein 3, I want to again remind you that this DNA sequence is exactly the same as the one above. Okay, but because I'm using Hein 3, I'm looking for AAGCTT. The cut is still made the same way. It's going to cut between the 2 adenines at the beginning. It's going to cut between the last 2 bases at the end, and it'll cut all the hydrogen bonds are in the middle. So again, another zigzag cut, but at this specific base sequence. So let's scan our DNA molecule to see if we have any AAGCTT. So here, AAGCTT. Because that sequence corresponds to Hindi 3, we will make our cuts between the first 2 adenines and the last 2 adenines, and then zigzag in between. Okay, so I have a small fragment of 4 on this end. Okay, now if you pause the video now, you can probably do the same thing, okay, with the other remaining sections. Figure out how many fragments there are, and then figure out how long each fragment is. Okay, so once you have done that, you can resume the video and see how you did. So this, oops. Sorry, having a little trouble here. Okay. Arg. There. So here, the first fragment had 4, okay, base pairs. The middle fragment has 44. Okay, so this one's a very large fragment, and then the last fragment has 11. Okay, so if you have the same number of fragments and the same number of bases, okay, then you have cut everything correctly. Now, it's a coincidence that we have the same number of fragments, okay, and even with the same number of fragments, we can tell that the size of those fragments are different. Therefore, HIND3 will create a different DNA pattern than if we simply just used equal R1. Now, in the world of genetics, if we have more than one restriction enzyme acting on the same molecule of DNA, we get a more unique and precise DNA fingerprint. Okay, because if I only use one restriction enzyme to cut DNA from multiple suspects, right, I might actually have, by chance, matching fingerprints. Okay, so the more restriction enzymes we use, okay, the more accurate our DNA fingerprints become. So if you scroll down or go down to the third section here, it's asking you to use both EcoR1 and HindD3 to cut the DNA molecule below. Okay, so again, you can pause the video here, look for the base sequences for EcoR1, make your cuts, okay, then go back and look for the base sequences for HindD3, and then make your cuts again. Once you do that, it will look something like this. Oops. Hold on, I can fix this. I just need to get rid of this text box. There we go. All right. So once you've made your cuts, in red, I've made the cuts for EcoR1, in blue for HindD3. So in total, I have 5 fragments. But because I have 5 fragments now, the fragments are typically smaller in size. So when I go back and I look at all the different fragments I have, I count up all the bases. The first fragment is 4 bases, the second fragment is 10, 19, 15, and 11. So because there's still different sizes, okay, the end result is when you use all these fragments, okay, from the 2 restriction enzymes to create your DNA fingerprint, that fingerprint will be much more accurate. Okay, so you will likely get an assignment where you have to make these cuts and then figure out whether or not a certain DNA sequence matches up with a certain suspect. So this kind of thing is typically used in forensic science, okay, which relies heavily upon this type of genetics. All right, so that's all the time I have for you today. Thank you for listening, and remember, I'm not going to have you memorize what the actual base sequences are. Okay, so these will be given to you. You just need to know how to cut them properly. All right, see you soon.